Whatever happened to predictability? The milkman, the paperboy, evening TV. Did I get delivered here? Somebody tell me, please. This whole world's confusing me. Clouds as mean as you've ever seen. Ain't a bird who knows you're true. Then a little voice inside you whisper, don't sell your dreams so soon. Everywhere you look, there's a heart, a hand to hold on to. Everywhere you look, there's a place of somebody who needs you. Everywhere you look, when you're lost out there and you're all alone, life is waiting to carry you home. Everywhere you
Today we are explaining the process of meiosis. Meiosis involves two special cells called gametes, which are the sperm and egg cell. The sperm is developed by the father and the egg is developed by the mother. Both of these cells are haploid cells, meaning they contain half of the amount of DNA compared to a diploid cell. Normal human diploid cells contain 46 chromosomes, and gametes or haploid cells contain 23 chromosomes, half of a diploid.
Although it sounds similar to mitosis, there are a few differences. In meiosis, there are two periods of division using the same stages from mitosis, prophase 1 to telophase 1, and then prophase 2 to telophase 2.
Let's start in meiosis 1, the first cycle of meiosis. Before the first phase of meiosis, in the cell's interphase, the chromosomes condense and pair up to create a tetrad. They also perform synapsis, where they connect together in a zipper-like protein structure connected at their chromosomes. Then, crossing over occurs where some of the non-sister chromosomes' DNA segments exchange with the other chromosome. The DNA segments split at the chiasmata and exchange to create a new, unique chromosome.
In prophase 1, the chromosomes finish the crossing over process, spindles form, and the nuclear envelope breaks down.
In metaphase 1, the homologous pairs of chromosomes arrange at the metaphase plate, and both chromatids are attached to kinetochore microtubules for each of the poles.
In anaphase 1, the proteins holding the chromosomes together break apart and the individual homologs separate, allowing them to move toward opposite poles guided by the spindle apparatus.
In telophase, each of the duplicated chromosomes are composed of two sister chromatids, each of which are a mixture of the paternal and maternal chromosomes that were provided by the sperm and egg cells. Cytokinesis occurs and splits the cell into two haploid daughter cells. In animals, a cleavage furrow is formed to split the cell into two.
Next is prophase 2 in meiosis 2. A spindle apparatus forms and the chromosomes move toward metaphase 2.
In metaphase 2, the chromosomes position at the metaphase plate and the kinetochores of the chromatids are attached to microtubules that come from opposite poles.
In anaphase 2, proteins break down and the sister chromatids separate at the centromere, moving towards opposite poles as individual chromosomes.
Lastly, in telophase 2 and cytokinesis, the chromosome begins condensing and cytokinesis forms, creating 4 haploid daughter cells at the end of meiotic division.
Meiosis creates variation between all organisms. Through the process of crossing over and the independent assortment of chromosomes, meiosis allows for genetic recombination of DNA, making each organism unique and special.